That's good.
Yeah.
So hi guys, my name is Joe. I'm going to speak quite briefly about a couple of the projects got going on, like through my PhD here in the University of Manchester with polyshields, where we're basically looking at how polyaromatic hydrocarbons affect the cardiovascular and liver sort of systems. So the one pH we're interested in really is called phenanthrene, but I'll get onto that in a bit. So yeah, first of all, I'm just going to speak a little bit more widely about air pollution, how it affects the cardiovascular system, and then touch on why we're interested in phenanthrene in particular, and the inflammation part, and sort of how that impacts the cardiovascular system as well. This sort of idea of systemic versus specific inflammation, which is something that we're quite keen to explore. And then I'll get onto a bit of the transcriptomics and gene expression before a little bit of histology finish. Okay, so as I'm sure you're all aware, we all know that air pollution affects cardiovascular health. I guess sort of people may tend to think about the lungs as sort of the primary effector, I guess, for the damage as a result of inhaling air pollution. But as you can see here, with about 800,000 excess deaths per year in Europe, with about half of these due to cardiovascular diseases, so ischemic heart disease and stroke, which is fundamentally a disease of the cardiovascular system as well. So quite similarly to Sam, we decided to do some modelling. So we combined census data, hospital admissions for ischemic heart disease, LSOA data in Greater Manchester, so LSOA is an area of about 1,500 people, indexes of deprivation, and then modelled air pollution data from DEFRA. So here's some sort of base level model data. It's not final, but for each of the 4 pollutants that we've modelled, we can see the increase in risk of death by ischemic heart disease per 1 microgram per meter cubed increase in concentration of the pollutant as an average. So for PM10, we're looking at 0.7%, PM2.5 at 0.14%. And where this sort of connects with the main part of the project really is that we're interested in polyaromatic hydrocarbons like phenanthrene, which are often bound to the surface or absorbed to the inner part of the PM10 or PM2.5 fractions of air pollution. So a little plug here for the recent paper we have published, where we basically did a meta-analysis for which shows sort of why we're interested in phenanthrene. So phenanthrene is essentially the largest or most abundant of pH in food, water, in the gas phase, and in the PM2.5 surface. And we've already done a lot of work focusing on the direct sort of electrical activity at the heart, which is affected like in a very serious way by phenanthrene, before just sort of cardiac arrhythmias and loads of different negative effects on cardiac ion potentials, which cause a really, really awful cardiac phenotype. But given that that's the case, we now want to sort of look into the more secondary downstream effects of phenanthrene, which one of the main ones, which Sam touched on as well, is inflammation as a result of exposure to phenanthrene. So we did a cytokine panel analysis where we essentially sent samples of liver, heart, and plasma from mice that have been exposed to phenanthrene to have the cytokines measured essentially. So I think there's 48 cytokines in the panel. So I've just picked one here. So one of the other ideas that we're quite interested in exploring is sort of where this inflammation is occurring. Because obviously you can have inflammation in the lungs when you inhale pollution, but also we know that these pollutants are translocating into the bloodstream and therefore will be reaching the cardiovascular system. So we're quite interested in seeing sort of whether this is a systemic immune response or a specific one that's occurring within the heart. So as you can see immediately, there's very little change in the heart between our low dose of phenanthrene and our high dose of phenanthrene. So then we look in the liver, which is the main immunological organ. And again, not massive effects. It is all non-significant because this was like a huge panel with just a small N number, which is sort of a pilot study. So then we also looked in the plasma. And you can sort of see, I guess, a trend which is sort of dose dependent in the liver and the plasma, which suggests that this is probably more systemic immune response that's occurring from the liver as a result of exposure to phenanthrene. So given that that's the case, one of the big things that we think, well, one of the big ways that we think this might be happening is from changes in gene expression. So to have a look at sort of how phenanthrene affects gene expression in the liver, we decided to do single nucleus RNA sequencing. So again, we've got 12 mice that we exposed daily for 6 weeks by injection with either the vehicle control, so that's no phenanthrene, a low dose of phenanthrene or a high dose of phenanthrene. So these are sort of representative of what you might be exposed to in a sort of low pollution environment, so I guess around here in Bristol, and then high dose is possibly what you might be exposed to in a really polluted city. So we've essentially got the mice, get the livers. Within the livers, we get the different cells out. Within the cells is the nucleus, and within the nucleus is the mRNA. So just to remind you what mRNA is, so this is from the translation of DNA when it is translated into RNA, and then it essentially affects, becomes proteins like IL-33, which I touched on in the previous slide. So by measuring the RNA, we can see what genes were being transcribed when the mice were harvested, essentially. So just a bit of actual information about what this entails. So about 7,000 nuclei put in with about 5,000 transcripts, which is RNA sections per nucleus, which identified about 2,500 genes per nucleus, which each compose about 350,000 reads, so bases, which is about 2.5 billion bases in total. So it's about 400 gigabytes of text of A, C, G, and T, I guess. So it's a very large data set. And so to accurately sort of visualize the relationships between the transcriptomes and the nuclei, we sort of need to use dimensionality reduction. So what is dimensionality reduction? So here's a woolly mammoth in 3D. So if you imagine that each one of the dots is a cell, I'm sure you can understand that there's really complicated multi-dimensional relationships between each of the dots in the mammoth. So if we reduce this down into a 2D format, you get something like this. So this obviously works quite well because we understand three-dimensional format of a woolly mammoth, whereas obviously our random selection of cells from within a mouse liver is not something that has sort of a recognized 3D structure. So this is essentially how it works. So unfortunately, my data isn't anywhere as satisfying as that. Instead, it looks like this, which isn't as exciting, but it's okay. So at the bottom here, we have in blue, mostly, so these are the control cells in blue. The nuclei from the low dose are in orange, and the nuclei from the high dose are in green. So each dot is representative of a transcriptome, of the whole transcriptome of each nucleus. So if you count the dots, it's about 7,000. So again, it's using 2D distance to display relationships between the transcriptomes. So sort of relationships between the different genes in the transcriptomes, I guess. So immediately, the one thing that stands out is this proportion of cells here, which are only found in mice that have been exposed to high dose phenanthrene. So quite clearly, the phenanthrene is causing something to change in the transcriptome that is creating a population of cells that don't exist in mice exposed to just the control, just the low dose phenanthrene. So then we can go through and look at the genes, the marker genes, and try and identify the different cell types within the 7,000 cells. So this is again sort of clustering done by algorithms and pipeline and stuff, which is really complicated. But essentially, we can identify cell types. So I mean, it's pretty basic analysis, but I need to do a lot more work on it. But essentially, I think we've got a subtype of macrophages in the high dose exposed mice that are differentiating sufficiently to be classed as a new cell type as a result of exposure to phenanthrene. Just there. Okay, so then we can look at some more of the genes. So within this big cluster here, we can look at what genes that are most overexpressed. So on the y-axis, an expression score, which is essentially log-fold change compared to control. So here are 5 genes, for example, which are all involved in pro-inflammatory processes. And they're overexpressed many fold times over compared to the control groups. So for example, CYP4A31 is a crucial role in modulating cardiovascular homeostasis, which is particularly interesting because although these are liver cells, quite clearly there are changes in genes that are having downstream effects in the cardiovascular system. And then we can do further analysis. So this is RNA velocity. So essentially you can examine the ratio of spliced and unspliced mRNA transcripts, and that basically gives you a prediction of the state of the cell. So when a transcript is spliced, it's ready to be turned into a protein essentially, and when it's unspliced, it's sort of not being turned into proteins. So it essentially gives you an idea of which direction certain cell types are going in. So for example, we can see this is, I think, the same cluster from before. So we're seeing differentiation of the ordinary couple of cells, which are the resident macrophages, into this unique subtype of cells. And the sort of thickness and length of the arrows denotes the speed of differentiation. So lots more to dig into really here, but interesting less. And then from histology perspective, so we've got liver sections, of course, from these mice, which we stained with H&E. And from the staining, we count cell size. So again, here we've got two different livers on the left with 50 cells in each, which are the blue dots. You can see possibly a small increase in size of the hepatocytes within the liver. But what's even more interesting is that the size of the nuclear area, so the size of the nucleus, does seem to be significantly increasing in a dose-dependent way. And then this adds up to show that the actual proportion of the cell that is taken up by the nucleus does seem to be a lot higher in the mice that have been exposed to the high dose of phenanthrene. So this is indicative of sort of DNA damage. So we've got some more work to do on that, see if we can sort of verify that. And then just finally, to finish off, can't really see it very well here, but we're going to be doing some immunohistochemical staining. So in blue here, stained on the nuclei within our liver sections. And in green is a marker for CD45, which is a marker which is on the cell surface of immune cells. So this is sort of a vein area. And the idea is that hopefully we're going to be able to see infiltration of immune cells in a more sort of pictorial way, which will sort of hopefully back up some of the other data stuff we've been doing. And this is something we can quantify as well, based on the intensity of colours and such. So just to summarise then, so the first part really is about the epidemiological model. So that sort of shows a more wide perspective, I guess, that there is a risk of increased ischemic heart disease due to increased pollution. And then we understand that this phenanthrene that is bound to a lot of these pollutants does induce inflammation in the liver and the plasma, which does have downstream effects on the cardiovascular system. And then we're seeing sort of the why, maybe one of the reasons why this is the case with the key liver cell populations sort of diverging as a result of exposure to phenanthrene, which again is going to be supported by the IHC and side cancer. And we just finished a 10-week exposure on 32 mice. So those 32 mice with phenanthrene every day for 10 weeks. So it's quite a lot of work. So we've got a whole new set of tissue basically. And we've got young mice, old mice, more N numbers. So hopefully we'll be able to sort of verify everything we've seen them. And that's pretty much everything. So, any questions?
Questions, please. Thanks. Fascinating talk, Rachel Smith from the UK Health Security Agency. I'm just wondering if you can take us through the sort of development of the sort of dose. How do you go from what we have inhaled to an intraperitoneal injection? How was that done? Would be fascinating.
Yeah, obviously it's not ideal to be using IP injection as a sort of proxy for inhalation. The equipment does exist to do inhalation experiments with animals, but it's not something that we have the capability to do. But obviously there's a lot more to it than that as well, as I'm sure you're aware. So anything that you inhale tends to sort of skip liver metabolism, as well as that being the case with IP injection. But in our new round of experiments, we've actually fed phenanthrene to the mice, so that's sort of getting metabolized in a different way. So yeah, I guess we just sort of try to pick doses that were as environmentally relevant. Obviously, it's not ideal, but hopefully we can do some exposure properly one day.
Raise their hand. Okay. Thank you very much.